What's going on, guys? It's Moon Daddy here, back with another video to teach you guys a little bit of something about cloning mature fruit bodies. I'm just kidding, guys. I don't even know. So, really, this is not the way I like to clone, guys, the way that I'm going to be showing you here right now. I like to generally clone plate pins, right? Sometimes your cubes won't give you plate pins that you want, or, and also, pans really never do. I've seen them a few times actually willingly produce them, but and I'm going to talk about this maybe another day, but I do know how to get them to actually fruit on your plate. But we're not talking about that right now because these are the fruit bodies that came from the bag grow that I was flexing on Instagram, where I grew a really nice heavy flush on an XLSB bag, these TTBVIs. And I've been working them for, over a year. Good year and a half, actually. And this is the F3. So from the spores that I got, that's considered the parents, right? That's my parent culture. So I've gone to spore 1, 2, 3 times since then, and they're just thriving now, without needing any air at all, really. So that's pretty cool. And I've never cloned this particular culture. I've just gone back to spore. I've tried to clone it, and they were contaminated, and I didn't have time to really go through and clean. These are very difficult little guys to clone if you're not used to it, guys. So I'm just going to explain to you how I'd like to clone, and this applies to larger mushrooms as well. With these, you just got to think smaller. And also, I'd like to point out before we get started, I always take a very small piece, as small as I possibly can, because that's all you need for that mycelium to reproduce vegetatively and grow into the culture that you're going to want, that's going to grow you the mushrooms that you want. Also, I'm going to tell you why we clone right now. It's for consistency. A lot of people think that just because you're cloning a mushroom that looks a certain way, that's going to be that mushroom. That's not the case at all, guys. These things can rewrite their entire genome in seconds if they want, just because they don't like the way your fart smells. Everything's environmental with these guys. Like, people talk about stability. Cubes especially are not stable. But mushrooms in general aren't stable. They do what they want, when they want, how they want to. The TTBVI generally is pretty stable though. So that said, just because you're cloning a fruit that looks a certain way does not mean that that's going to be the fruits that grow the next time, unless you have an actual mutation. So the chances of that are pretty rare considering how many mushrooms you're actually growing till you actually see a mutation, okay? If you have a really nice culture and you see a mushroom that you like and you clone it, chances are you changed that culture and it's not going to be the same anymore. So are you willing to make that? People always tell me on Reddit all the time, I'll post a picture of some fruit or a fruit and they, type out, clone it, ! And it's like, why would I clone this? Look at what it's giving me. Look at what this culture is giving me. I've had horrible things happen, like completely lose culture. They just go to shit one day because you decide to keep cloning, and it's just not necessary. I don't know what it is about it that causes that to happen, at least in my experience. But a lot of times, man, it just ends up ruining the vigor in the culture. I don't know if it's because. So I'm going to put it to you like this. This is the way I see it. Okay, just because you isolate, say, an albino, that doesn't mean that that's a new isolate. So the two spores that meet and they clamp and they make a mushroom, and then that grows, and then you grow more mushrooms, you clone it again, that's still the same organism. That's still the same dikaryon, the two monokaryons that decided to hold hands, build a mushroom, and make a nice little shag and shack up there in the gills to produce babies, right? That's the whole purpose of those two mons, clamping and holding hands, building a house, is to reproduce inside that house, okay? You can clone it 10 times. It's still that same isolate. But what's happening? Say you end up cloning an albino 3 generations in. It's not that you have cloned a new isolate and that isolate is albino. It's that isolate at that moment is expressing those genes that are the albino genes, that are the white genes. At any moment, it could revert or just change back to the normal color that it was. I look at it like this. So I imagine myself splitting from the moment that I'm at into two different timelines. So there's 2 me living on 2 different timelines. One of me decides to take steroids, eat 6,000 calories a day, work out for 4 hours a day, and bodybuild for 6 months, okay? And the other Tony, or the other Moon Daddy, he decides that he's going to go into basically pretty much silent darkness for 6 months and meditate and fast like Gandhi. Very little sustenance to keep him alive. Okay. Just basically living off the vibes of the universe. Okay. And he's meditating and he's just doing his thing in the dark room. And then eventually those timelines merge again at 6 months and they see each other. The genes that I'm expressing at two different timelines, just because I'm expressing those two different genes doesn't mean that I'm different. I'm still the same exact human with the exact same DNA. Okay. Either one of them could go to a crime scene and leave DNA behind after doing some crazy shit and frame the other one because they're the same person. They're clones of each other. But they don't look or act anything alike. And we know why, because of environmental factors that are changing the way our bodies adapt and the way that we look and the way that we feel. Everything about our environment determines everything about us. So, that said, that's why you see different shapes and colors of mushrooms. Okay? It's not that you've isolated an albino gene. It's that the traits that express the albino that are already there are being expressed stronger in that moment. Okay. So the way I like to clone, guys, spray it down with hydrogen peroxide. Obviously, you guys know me. I love the H2O2. Kill all the germs on the outside. Do not cut your stipe open. Simply peel it open with your fingers like this. Okay. And when you do this, it keeps, instead of using a knife would push all the bad stuff on the outside right into the inside, and that's what I want to clone. I don't want the bad stuff pushed inside on the stuff I want to clone. Take a tiny piece. The reason why we take a tiny piece is because the smaller the piece, the less likely it is to have contamination. Okay, and guys, the inside of these fruits, inside of fruits aren't sterile. They're not supposed to be sterile. Granted, they're very clean if you're very clean with your practice indoors, but just the way we have a biome inside of us, they aren't sterile either. So it's insane to me that a lot of people think that inside of mushrooms is sterile and they're cloning fruits from the third flush. And they think that they're not piggybacking all sorts of nasty shit that's going to eventually bite them in the ass, at least my experience. But you see that, you basically peel it back like a banana, right? So, right, you see those little fibers. Look at this. Oh, it's beautiful. Yeah. Oh, yeah. Check it out, guys. Man, I get talking. I can talk. Look at that. Just like that. See those fibers? That's the good shit. That's what you want. You want to get a little tiny fragmented fibrous piece of that wasn't on the outside of that mushroom and transfer it. I mean, the reason why we like smaller, guys, is because the less room it takes, the less problems. The more room that there is, the more problems. That's why I don't grow in a tent. That's why I like to grow in a fruiting chamber that's automated, because it's less space and it's less problems. It's less things I have to worry about that I have to control. It just makes sense to make it easier. Simplify things. Take little tiny itty bitty pieces and they'll grow. Okay. So the next thing I want to talk about is pretty important also, guys. Let me see what's going on. Let me see what's going on, guys. Yeah, guys, this is important. The next thing that I'm going to talk about, and that is making sure that your blade or your tweezers or whatever it is you're using, I normally like to use tweezers, make sure they're all the way cooled down, because like I said, we're taking just tiny samples of this tissue from the inside of these fruit bodies, and if it's hot, it's going to just kill it. So you're going to waste it. But all you need is a little tiny piece, guys, like I'm going to show you right here. And that's perfect for your clones. You're not going to contaminate. You might still contaminate, but it'll probably be deal withable. I mean, after all these plates were grown out so far, one of them was just green. All of the other ones were just fine. So during one of these transfers, I definitely messed up somewhere, but that's fine. That's why you do more than one, okay? Yeah, like I said, guys, you want to make sure that you're all the way cooled down and that you're clean and you're ready to go. I don't know. I'm tired. Anyways, Make sure you're clean and ready to go, guys. That's what you want to do. It's very important. And make sure that your shit's cooled down, all right? So these are, I got these off Amazon. They're actually jewelry holders, like bead holders. They're dope. They're polypropylene. They screw on tight. I use them for long-term storage, guys. And for my master slants or plates, cultures, right? So I can always go back to them and stretch them. That's my one that I go to instead of just like always wasting a new plate. Sometimes you got to do that anyways though, but these screw on tight. They seem to stay good in the fridge for a long time, and they save me a lot of space, like literally 5 times the space. It's pretty tight, so stoked about that. Check out how small this piece is I grabbed though, guys. It's a little tiny fibrous piece on the inside. It's all you need. You don't even need this much. You literally only need 1 dikaryon. That's tiny, right? 1 cell. Yeah, that would actually be 2 cells, 2 haploids, right? It's not a diploid until karyogamy occurs up in the gills after they build their house, after the 2 mons, hold hands, build their house, and they go shack up and do the nasty, do a little exchange of genetic information up there. So, this is 2 haploids. But yeah, just little tiny piece. I just have my tweezers right here that were actually broken in half, and they work great. They make it really easy for me. And yeah, we'll watch me do it a couple more times right here, and then I'll let you guys go. But yeah, I really I just appreciate you guys, man. You guys are awesome. I really, I never thought that this YouTube would be I would have so many followers so fast. Like, I really haven't promoted it at all, so that's awesome, and I appreciate you guys and your reciprocation of appreciation for the knowledge that I give. And yeah, it's just one of those things, man. This community is badass. Even if you're having a bad encounter, it at least for me, it inspires me to be better. And good encounters, obviously, and I can see that everybody's inspiring everybody, and we're asking questions and we're pushing the envelope. We're doing things that 20, 30 years ago, 10 years ago, 5 years ago, they said that we would never be able to do because you can't do it. And look what we're doing. It's awesome. That's why this it's like a sport, right? The sport's evolved. I'm just kidding. This hobby, I don't even know what to call it. Mycology has evolved and is evolving into something that's just making it easier and easier and easier. Like the way that it's done now, like I grew these in a freaking bag, in an XLS bag, guys. That's awesome. We did it. We freaking did it. And I wouldn't have done it if I wasn't trying to possibly show you guys how to do it, And I hope that it inspires you guys to go and create and come up with new ideas and new things and help people and, just make the world a better place. So with that said, let's check out the little tiny fibrous tissue sample from this dope ass TTBVI. Let's watch me put it on a plate. What just happened? How did that happen? Anyways, there it is. How did that happen? I don't know. Who cares? Check it out. Oh, no, we already did that one. Let's do another one. Since we're here. Come on, Tone. Come on, Moon. What you doing, Big Papa? Come on, let's go. Oh, there it is. Dun, 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 dun. Cool. Take that little tiny fibrous piece. Okay, I guess that's where it ends, right? Anyways, good enough, guys. Freaking love you guys, and you get the idea. That's how I clone. Do the same thing with your larger fruits. This is way harder, so I think that you guys get the gist of it, and I hope that you guys have an awesome rest of the month. Hopefully I can drop a new video sooner than that, but till then, much love, guys.